Hello, everybody, and welcome to another issue of the NIPB Education Committee webinar series for our fall series. Today, I have the pleasure of introducing Dr. Salvador Gezan. He's a statistical genetics consultant at VSN International. Today's presentation title is Parentage Reconstruction, a powerful breeding tool to exploit the genomics era. In this webinar, we will explore some of our procedures with the use of genomic information in different algorithms to check, assess, and reconstruct pedigrees, and to identify duplicates and sibships. Further elements associated on how useful this is for breeding strategies will be discussed. So over to you, Salvador.
Okay, thank you very much. Can you hear me well then?
Yes.
Okay, good. So I'm going to go over particularly one specific R library that we've been building, but more than showing you the specific library, I want to show you a few of the potential uses and the needs, and even how to kind of consider this parentage reconstruction within the strategy from a breeding program. I have worked with salmon, trees, and different agricultural crops, and even animals here and there that make kind of relevant the use of the parental reconstructions on different strategies. So I'm going to just go a little bit of an overview, then I'll jump into the demo and come back with a few more slides. So I'll try to stick to the hour, and hopefully I'll have a few more minutes towards the end to give you the opportunity to ask me questions and so on. But you can always send me emails or a note or hear something to ask things. Okay, so the first thing is when we talk about the pedigree, or sometimes we use the word parental reconstruction, we refer to the use of genomic information, some form of genomic information, and this could be a small panel, a large panel. I'm thinking mostly on the cases where we have already some data that we use for genomic prediction. So I'm thinking about 3,000 to 5,000 SNPs, all the way up to 50 or 100,000 SNPs. So we use this genomic information nowadays for most of the genomic predictions and for GWAS analysis, but we can also use this sort of information for reconstructing the pedigree. And there is many uses for the reconstruction of the pedigree. We can, for example, I name a few here: verify the historical pedigree and crosses performed, more like a quality check. To check the quality of the molecular matrix, we might have a molecular matrix and we more or less know what to expect from there. If we can pair that up with a pedigree, we'll be able to say, okay, there is some issues here or there. To assess the quality of both the pedigree and the genomic relationship matrix, again, sometimes you have duplicate individuals, sometimes you have offspring from a parent, and then you obviously expect that the genomic matrix will accuse the same things, and sometimes it does, and sometimes it doesn't, and then tells you a little bit of what's going on. To complement our typical breeding analysis, in this case, we can use GBLAP, but we can also use the single-step GBLAP, which makes use of this age matrix, which again, it's a combination of pedigree-based matrix and genomic-based matrix, and allows us to combine information from individuals that are not genotype. And I'll talk more about it. And again, once more, we need to kind of check this pedigree. And then we can incorporate some of the uses of this pedigree reconstruction as part of our breeding strategy, maybe sampling some individuals and so on. I'll talk a few more things about this later. You're probably familiar with the pedigree-based relationship matrix. Actually, I noticed probably once with the emergence of the genomic selection and the use of the animal model to fit a GBLAP model, it became more relevant to consider the pedigree. Some breeding programs have started to reconstruct their pedigree or actually to collect their pedigree. It's a historical pedigree. But in animal breeding and in tree breeding, we have been used pedigree for 50 years or more. And then the way we calculate it is we just provide a pedigree file where we have the individual, the male and female, if we know it. If we don't know it, we put a 0 or an NA, and we use this to construct in this case, pedigree-based relationship matrix that gives me the probability that two individuals are related. There's other definitions of it, but you can see on the elements on the diagonal, you have one, and anything bigger than one denotes some level of inbreeding, and the relationship between individuals, parent, offspring is 0.5, full Sibs is 0.5, half Sibs is 0.25. So it's very standard, and it relates everything and allows me to predict breeding values for individuals that have not been tested, which is pretty much what genomic selection or genomic prediction does in the case of the GWAS. So we can move from this pedigree-based matrix where we have a few values. If we have a SNP data, we obviously will calculate a GA matrix, sometimes called observed relationship matrix, genomic matrix, GRM. It has very many different names, which is going to be an estimation of that relationship. Again, it's not going to be exactly 1, it's not going to be exactly 0, because it's sampling the SNPs and calculating this. There's many methods. The most common is the band grading to use, and that GA matrix has some caveats. It has to be invertible in order to use into a mixed model approach. So sometimes we have to do a bit of tricks to make it invertible. And you have to be also careful that that GA matrix sometimes will be a bit unstable. That the reason why it cannot be invertible, maybe because we have duplicate individuals, or because some individuals have too many missing values that create some issues when you do some imputation with poor quality imputation, and so on. So the GA matrix often has to be assessed in order to fit our GBLAB models. And we do eliminate duplicates, we do quality control of the markers, we do different types of filtering, pruning of markers, and so on. And also there's 3 tricks that are kind of relevant. One of them is bending the matrix, so try to make some of the eigenvalues slightly positive. The other one is to blend the matrix, which means we average the genomic matrix against the relationship matrix, for which one we need to have a recent relationship matrix. And that kind of makes it a bit more stable and away from extremes. And the other one is very important, is alignment. The GA matrix is being calculated based on the P's and the Q's from your reference population, but the A matrix has all the historical inbreeding that you don't have necessarily on the GA. So sometimes it's good to move the GA towards the A. Another reason why we need sometimes the A to do sort of those alignments and fixing of the GA. So I'll use 2 libraries from the company I work with, VSN, and I've been heavily involved in them. One of them is the ASR Genomics library that does some quality control, SNP pruning and things. You can do this in many other software, Plink and so on. But one of the things is that we have a routine to generate the inverse and the G matrix. And if there's any problems with the inverse, you can use some tune-up functions that I mentioned before. So I'll be using some of these to kind of filter and get the G matrix. And I get some diagnostics on my matrices, because those are very important also to assess what's going on. And then a library that we haven't released yet, but again, it's a library for parentage reconstruction. We call it ASR Parentage. Now, there is many other libraries that do parentage reconstruction, and I was just writing a name of some of them. You probably heard about Sequoia, which is quite good. You have France, Pet Builder, HS Face, and so on. I have used Sequoia before. Which you have other alternatives to use. Now, in this library, the way I'm going to kind of check my data is first focus on the duplicates. And notice that I have functions for duplicates using the GRM matrix. So you calculate the Van rating and explore the GRM matrix, or using the molecular markers, which sometimes I call the M matrix. And then you can check individuals by pairing up which ones are similar to each other. There is slightly algorithms to use that I'll explain in a little bit. And then the other functions are to find the parental cross. So you have the kids, and then you have a group of potential parents, and you try to pair or you have to take the individual kid, and with all the possible parent pairs that you might have, it might tell you, oh, this is your more likely pair. The other option is just to find one of the parents. Some breeding programs, you only know one of the parents, or you only genotype one of them. And sometimes, again, it's useful to verify things. So if you get assigned to one parent and then get assigned to another parent, and those two are males, then that's going to be a bit of a problem, really, and to think that that's your specific cross. And a few more things that I will go. So if you're interested in this library, please send me an email. And we have a beta version to use and to explore. Now, the algorithms are based on something that is called inclusion-exclusion. You have pedigree reconstruction software and libraries that use a likelihood-based approach with probabilities that you see this given that you have the other individuals and so on. This is just more like a, let's say, bit of a brute force comparison. So, for example, here I'm looking for duplicates, individual A and individual B, and you have their markers assess and 0, 1, 2, where 0 means AA, 1 means AC, and 2 means CC. So the typical marker set up for our genomic selection. And again, this library works best when you have something like 3,000, I will say between 700 to 3,000 SNPs. You can filter down if you have a panel of 50,000 and just sample every 10th or every 20th, depending on what you want, and verify it on different combinations. So in this case, I'm looking at what are the difference between these two individuals, and there's no difference here. There's one difference, which is one allele that is different, no difference, no difference, and there is another one that has two alleles difference. So you want to find a list of pairs of individuals on which the sum of this distance is an absolute sum. It's the smallest, so I call it genetic distance. When you have parent pairs, the logic is a bit different. You have an individual, but then you have a pool of parents, and in the pool of parents, you make every possible cross between the pool of parents that you have. You might be missing some. Once you made that pool of crosses, so you can see here 2, 2, 1, 0, 2, 2, 1, 1, what we focus on those SNPs that are going to provide homozygous alleles for their offspring. So a 2, 2 gives a 2, 2, but a 1, 0, we don't know what it is going to be. It could be a 0 or a 1. So we don't deal with that one. We eliminate that one because we're dealing with large numbers of SNPs. We still have plenty of information. Again, a 2, 2, and then we have a 2, 0. The only outcome from this cross of these two SNPs, this SNP for these two parents is a 1. So you inherit an A and then you inherit a C. So you have to only be AC, and that's going to be the outcome. And then you compare that cross with all the individuals that you have as potential offspring, and then you calculate again once more a genetic distance, which is going to be again once more the sum of these differences. So it's a very simple algorithm computationally. We did it in a parallel way, so it runs relatively quickly. So let me just go to the demo and show you a few things. Again, this is not necessarily the only package you can use. I'm just going to give you the flavor of it. And then once I show you a few things, I'll give you a bit more about how you can use some of this into your breeding programs and some of your checking with this or with another package. So I'm going to use a data set from Apple. This is 247 genotypes, and I think I have 2,828 markers. So it's a bit less than 3,000. The first thing I'm going to do is I'm going to do a bit of a filtering of this. So you eliminate the ones that have some levels of missing, but I'm being a bit more strict with the minor allele frequency here than with other things because very low allele frequencies are more likely to be confounded with what it is a bit of genotyping error. So a 10% MAF gives me still 2,330 markers. And then some other ones were eliminated for other reasons and so on. So let me just focus on that. And I have my clean matrix here. Now I add this line here in case you find that you have too many SNPs. You can sample directly from this. You can also do a pruning, like a genetic pruning, where you use the LD between SNPs. In some other cases, depending on your breeding strategy, you might just have 200 SNPs. That's what you're going to use to do this, so you don't need to do any sampling. You might also want to do it by chromosomes. So there's different ways that you can have. But my logic, again, is to use something around 3,000 SNPs. This is the calculation of the math behind. It's a genetic distance between the difference, the sum of the difference divided by the number of alleles. So it gives me the percentage of alleles that differ between two potential individuals, okay? So I'm just going to run this duplicate function. You provide your N matrix, and you provide a threshold. And this is threshold is saying, give me back any pair of individuals that have less than 11% difference in the genetic distance. And this one takes a little bit of time depending on how many individuals you have, but it compares every single potential pair of individuals. It's using 7 cores, and it tells me that I have potentially 9 individuals that might be duplicates. And this is where this interpretation becomes more interesting. So you have this individual against that individual, and this is actually real data from a publication. The weight denotes the number of SNPs I have used to make that comparison, and the distance is 1. That means there is only 1 allele difference between these two individuals, which corresponds to a 0.02%. So clearly these two are the same individuals. And then you have 5, 5, 7, and then you have another one with just 1 difference. So these individuals really, these 5 pairs really are the only individuals that are identical. So I have to do something with them. Do I drop them? Do I regenotype them to assess which one is the one that is incorrect? Maybe I have some reference, but you can see here they are from different families. The 3 numbers here corresponds to the families. So it is difficult. This is probably an issue on the management of the DNA sample. I wouldn't say contamination or something. Something happened. Now, then you have the individuals that have like around 9 to 10%. And this, so you see these individuals have a lot of SNPs different. A lot of alleles that are different, 9 to 10%. So these clearly are not individuals that are duplicates, and actually they belong to the same family. So they are likely to be full sibs, and they may even share a grandparent in common. So they have a higher relationship. So that's good. I can also use the genomic matrix. Notice that I use the function duplicates SNPs. I can also use the genomic matrix, which I'm going to calculate with my filter matrix. Bear in mind that this genomic matrix sometimes has a little bit of bias because the P's and the Q's you use to calculate. So I have 0.97 and so on. And I can run this with the GRM. And in reality, what it's going to be is going to look for any relationship that is around 0.9 or higher. And it found 5 combinations. And these are the genetic relationship of these individuals according to the GRM. And we have actually the same 5 pairs that were found before. I do personally recommend the use of the SNP to do this. Now, how do we define the threshold? We can play and assess. We can also do a bit of we add a function to do a little bit of a simulation, sort of like empirical simulation. So you can create the same number of SNPs with a percentage of missing values, with percentage of errors, how many samples you're going to do. And it's going to run several pairs of individuals that should be copies and pair of individuals that shouldn't be copies. And then at the end, it tells you that some combinations were suggested as duplicates when we know there is no duplicates. And this is where it tells you the threshold to make the separation. So in this particular data set, a threshold of 3% or somewhere between 2.4 and 3 will be good enough to provide me a difference when I have an error of 2.5% on the genotyping. Again, it's just to give you an idea. So it's not like, okay. Now that was a data set for duplicates. We can also, I don't have the parent's genotype, but I have another data set, which is a salmon experiment where I have the parent's genotype and the kid's genotype. So I'm going to load this data set. Again, I can make all of this available if you need it. Just shoot me an email. This one has 2,000 SNPs and 624 individuals. It's already been filtering from close to 50,000. So it's just a little bit of a subset. Now, these 624, around 90, 92, 94 individuals are the parents that are being genotyped, and the rest are kids. So I have 500 kids, more or less, a little bit more than 500. And I'm just pulling the actual data. And then you can see a little bit that what I have here is I have the individual. This is the first few, the parents. This L2F are the female, L2M is the male, and we have the sex. And I have this number which corresponds to the generation. So it's important to specify which one is generation 1 and which one is generation 2. I'm showing you now a portion of the offsprings. This is the name of the offspring. It's a male. Irrelevant to know the sex of the offspring. It is relevant to know the sex of the parents. If you don't know the sex of the parents, you just put unknown, and it's going to make every possible parent. If you put the sex of the parent, it's only going to look for males crossed with females, and not females with females and males with males. So it reduces the complexity of the problem. And notice that we have a number 2. And then I have all the SNPs to the side that I already filtered. So again, this is just to tell you a little bit that I have in generation 1, I have 80, almost 90 parents, and all the rest are kids on both of the sexes. Okay. So I'm going to extract some, I'm going to just pull the molecular matrix, and I'm going to, the I data, actually, it's good to look at the I data because it is the one that defines the structure. It's the ID of the parent or kid, the sex, if you know it, otherwise unknown, and generation 1 and 2. So again, what it's going to try to do is going to take the generation 1, create all the possible pairs of parents, and then take those possible pairs and go one by one of the kids to see where do they fit. So before I do that, I'm going to do once more a little bit of a filtering of the data. And there's not much to do. 2 markers were removed from some, so there's not much filtering because I already kind of did it. And now I'm running the function duplicates just before I go here, because obviously the duplicates are going to be accused to be kids from the same groups of parents, but they are duplicates, so they might be a bit confusing. So if you run the function duplicates, in this case, we're not going to find anything. So it's comparing all the parents, all the kids and parents, whatever they are, without caring too much about what happens. So it accuses, I put a threshold of 12, which is quite high. But you can see here, out of all the SNPs, 452 alleles are different, and then that's 11%. So these 2 individuals here are actually related from the same family, but they're not duplicates. So that's a good thing. So I can proceed to run my pair cross, which is the function pair cross for the SNPs. What do I need is I need to specify the molecular matrix. I need to specify my individual data, which is the I data I show you already with the individuals, the sex, and the generation, indicate which columns are which, and then indicate a threshold, which arbitrarily I pick a threshold of 5%. Now, because the way it works is going to compare every kid against every potential pair. And remember, a pair of parents is only going to look when you have a consistent offspring. So if a parent is 2 and the other one is 2, it's only going to be 2s, and that's okay. If a parent is 2 and the other one is 1, it's going to drop that allele, which means different comparisons, we have different precisions, okay? Now you can see here there is 1,769 possible crosses, and every single individual from the offspring is compared 1,769 times. So this is almost 1 million comparisons. And then was done in 7 cores. And what it's telling me here is I have 531 offsprings that were assigned to at least 1 pair. It might be that they're assigned to more than 1 pair according to this threshold. But this is where things start to become a little bit more interesting. And 3 were not assigned to those parents. By the way, if they're not assigned to a parent, it could be that maybe it's a contamination and the male or the female are other ones. maybe we couldn't genotype one of the parents. And in the list of 90 parents that I have, I should really have 92 or 95, but some of them were not assigned. Maybe I can find out which one is the female or which one is the male parent, it's one of the parents of that individual, and continue constructing this. So this is the individuals that were not assigned. And this is, I'm just going to show you the database, how it looks a little bit. So this is the first individual, and the first individual, if you can see here, it's a bit small, but the first individual 0001 was assigned to this pair, and it has weight of 596, the SNPs that were compared, and only one is different, and this 0.08% of difference on distance. So in reality, this individual, that is the two parents. The other two pairs that are assigned is probably the mom and an uncle, or the dad and an aunt. So it's not really finding the right, and it's a lot more errors, and 2%, 5%. So this is quite easy to assess. Notice this is 0% to a 2% already denotes an error, 1.5% denotes an error, and so on. So we can look. Some individuals are going to have a lot of assignations. So what we really need to do, you see it says one individual, only one pair, 18 individuals to one pairs, but there's a lot to more. So what I'm going to do is I can change the threshold to 1%. Again, we can play a little bit with different percentages. And again, it's going to do the same set of comparisons. It's going to take around a little bit of time, but it's going to filter down this pedigree. And that's the lines that we want to use. So let's just wait a little bit. I wonder if there's any questions so far. Maybe somebody wants to make a question if they are allowed. No. Okay. Well, there'll be a bit more time for questions. So this should be finished in a couple of seconds. And we'll give it a little bit of a minute. One thing while we wait for that is we can also calculate. I have a function that calculates the threshold for the parents. I'm not going to do it here, but it allows to, again, to have a more objective way to define the threshold. Let me show you the output. It says 520 individuals were assigned to at least one pair, 14 were assigned to 14 were not assigned to any parent, so there is a few. And notice here, 459 kids were assigned to one parent, 50 were assigned to two parents, 11 to three. So what we can do is we can export this data frame, and you can see, and some individuals, I think, for example, we might have, I remember 28, individual number 28 has two potential parents. You have 2 and 11, so you have 0.16 and 0.85% genetic distance. So it's more likely that the first one is the actual parent pair rather than the second one. So you can drop that line. Again, the thresholds are not really too different, and that's going to be easier or harder depending on your population. Notice this one is 155, has 3 potential pairs, and one of them is with only one error, the other one with 8 and 13. So you can see here, it's very likely that this first one is not going to be perfect because you have genotyping errors and so on, but it's going to give you a little bit of that idea. Okay, so I can define some threshold and do some simulations similarly as I did it with the duplicates to get a better threshold. And this is telling me that a threshold of 1.5% is the one that is going to give me possibly the best combination. You can play with this in different ways again, and I'll show you. I'll show you that into a little bit. Now, while we're waiting for this, we have also the function for single parent. And this is a little bit easier because in reality, you don't have to construct the crosses. You just compare against every single parent. And actually, you can see how consistent is the parent pair. Now, it's relevant in some programs where, like, for example, in trees where we have open pollinated, we know the female, we don't know the male, and the male might come from outside of the orchard or might come within the orchard. So we might want to do it first, the full parent pair, and try to see what we can reconstruct for the full parent pair. But again, there will always be pollen from outside, and then we can compare with a single parent. So in this particular case, we found 14 individuals, again, that are not assigned to any of the parent pairs, and there's more options to assess through the rest. For the single parent pair, which, as I said, it is easier, you use the same individual data. You only need, in this particular case, the generation and the sex, but it's irrelevant, the sex. And so, but you can provide the same data as before. I use a threshold of 10. Again, there's ways to assess what happened. 534 offspring were assigned to at least one parent, so I am able to find at least one of them. So this is how it looks. You have 452 assigned to this female with 130, which is probably incorrect. Let's look at the full database. And I still have some time, so let me, here it's sorted by ID, and you can see this same individual was assigned to a female and a male, and these are the distances between a female and male. Number 4 was assigned to this female, but also was assigned to this female, which is obviously incorrect, but it doesn't have a male. So this female is probably an aunt, and there's no male. So what we can do is now we are ready to create the pedigree for this individual. We put the female, and the male will have to put a 0 because we don't have it. Okay, so as you can see, I'm slowly reconstructing the pedigree and checking things. Okay, I can use a more strict threshold. I can play with what is more reasonable, and so on. And I can also pair the two databases, just kind of like to have something more aligned to see if the individuals the parents that were assigned as parents in the pair and the parents that were assigned parents as single, they do match or don't match. Okay, again, this is all sort of like computational things to simplify the process of finding the pedigree. Okay, so I'm just going to show you a little bit what it does. It tells you the offspring, the parent pair, and then it says when one was found for which one were all the potential parents that were found for that individual on the single parent. So you can see here for that individual, they find 276, which one of the parents, and 2151. But for the second individual, which is 5, assigned this parent pair, 1741, which was defined before, but also 2171, which was defined before, and 23306. So there's one potential parent that seems to be extra, and some of them have more extras. Again, just to show you that you can do a little bit of pairing between these and see what's going on and what are the differences and so on. So let me go back to my slides because there's a few things I want to mention. And so how do we want to use this reconstruction of pedigree, again, with this package or with any other package? So this is, I'm just going independent on whatever we have. I guess. So the first thing is, let's assume is using this reconstruction under where you still are going to focus on a pedigree-based strategy. So you're still going to use your pedigree analysis for your data as you have done historically, thinking more like in some animal breeding or in some forestry. So you're still going to be there as done it, so as you have done it. But now you have small panels of maybe 200 SNPs, maybe 500 SNPs, or microsatellites that you can use and reconstruct in a different way. So to use your reconstruction of your pedigree, just say, oh, I'm going to use this to know the parents and so on. It is relatively cheap, and you can also use it with other uses of the same SNPs, like when you're looking for marker-assisted selection for some things. So it's cheap and allows you to continue using your pedigree base and your animal model, but allows you to reconstruct your pedigree or check things without having to worry too much about who crossed with what. So it improves the integrity of the data. Now, there's many breeding strategies that I'm going to mention later that use sort of this approach. Then something about the quality control of the breeding. I have done crosses, and it's very common that you do the crosses and then you have your individuals that are going to go into the field, and then you genotype them to verify that you really did the cross that you expected to do. Sometimes you have a group of individuals from the same family, so you can get 1 or 2 individuals as samples to check. But sometimes you're in the field, and I've seen this in forestry, where your experiment is being established and then you get small DNA samples, small panels and DNA sample, and you're going to verify what you wrote and what you expected that you did in there. So it allows me to estimate levels of contamination, improve my pedigree, because you're going to find errors there, or you're going to find you have pollen contamination, etc. And it allows you for obviously a better control of relatedness of inbreeding, because if you are getting the wrong pedigree, you might be assuming some individuals are unrelated when in reality they are related, and so on. So this is often based again on a small number of SNPs, but sometimes you're doing it early on the experiment, and you may have your full panel of 50,000 SNPs, and then you do it early on. So then we have the other option, which is genotyping quality control. I like to think about this more like I have my M matrix. I'm going to get ready to do my genomic selection. And then I need to do some quality control on the quality of the marker data. And so if you have a pedigree, you can pair that one with a pedigree. Okay. So it allows me to fix things, thinking more about the duplicates. So find the potential duplicates and what they are duplicates. And sometimes we expect the duplicates because we send the same individuals to genotype at different time points as a check. But if we don't find them, it's a problem. But if you have the pedigree, you can just also use some verifications. You know, some of the parents you send are related. Let's see if they're still related. If it's not, what's going on? And you can find all those mistakes. So it's more about focusing on the M matrix and how it is. Now, the GRM quality control that I have there is more associated that I'm going to calculate a genomic relationship matrix, and I have a pedigree. Is the genomic relationship matrix accusing the same pedigree that I have? So in that case, I can use directly the molecular marker as I just shown to reconstruct, or we can use the GRM and compare with a potential pedigree and see what are. So for example, what you can do is you can just take your molecular data and reconstruct your pedigree again and pair that with what you have as a pedigree. And if there's differences, you say, oh, is the pedigree incorrect, or it's the genomic matrix with some issues. I do recommend this because even that you're not going to use the pedigree, there might be errors on the samples. There might be individuals that were, again, duplicates, or individuals that were declared unrelated, but your pedigree is saying they are related. So maybe what happens is they take the DNA sample from another individual or got something mixed up. I have seen this before. So, and then also the A matrix is going to help me to do some tuning. The blending, the alignment are very important in order to improve the quality of the GRM and make it invertible. So you want to do your blending with an A matrix that you trust. So if it's not really doing anything, then you don't necessarily do it. Something similar with the alignment, where you shift the diagonals on the off diagonals to match the A. And in general, it's going to just give you more quality, less issues, convergence issues when you fit your models and so on. And this is also for the Bayesian models. Again, garbage in, garbage out. So it allows you to do that. It does take time. And probably one of the most interesting uses of pedigree reconstruction from a G matrix is to check that really matches the A matrix that you are going to have from your historical pedigree. And, you know, if you're aware of the single-step GBLAB, uses all the individuals that are being genotyped uses the portion of that G matrix, and the individuals that only have pedigree information is going to use that portion of the A matrix, and trying to alignment and make it in a good agreement. And you can see here some of the operations that occur behind. And it is important that what the genomic relationship matrix is, these two individuals have a relationship of 0.12. Well, hopefully that's being accused also in the pedigree matrix. Otherwise, you're forcing things in one way or the other, and you get really strange things. Okay. So for example, the more typical case is you have two individuals that are highly related. That means the parents have to be related, but the parents are unrelated and the grandparents are related. So when you do this system, you're going to force relationship of the parents and the grandparents. And what's going on there, we don't know. So the instability of those agreements, it will make the H matrix more unstable, maybe not invertible, and obviously it's not going to make the most benefit of the single step GWAS as we wanted. Okay, so those are a few of the things. I'm just going to mention two more things. I still have a few minutes. Sometimes we have no way to define the parents because the parents are not genotyped, but we still can find sibships. Sibships most probably means more like full Sibs or half Sibs, groups of individuals. In some approaches, you can find the sibship and say, yes, the male one and female one for this family, for the other family, male 2, female 2, and so on. And at least that gives you an approximation of the relationship, and you can combine that information maybe with whatever genomic matrix you have, or maybe just to verify that things are happening. So I'm going to use the a much larger set of genotypic data from salmon, where I only have offsprings. So I have the fish, I have a number of markers, 17,000 markers, and 1,481 individuals. I'm just making a bit of a filtering, but my objective here is to calculate a genomic relationship matrix and do some form of cluster analysis on the genomic relationship matrix to calculate sibships. Okay, I'm just going to calculate the GRM matrix using the band rating based on this molecular data that was filtered, which is actually only 11,000 SNPs. So a large group of SNPs were dropped, almost 6,000. And let's see where we get. It shouldn't take too long. And then I'm going to use this shift here, GRM. What it's going to do is going to create a cluster analysis, and it's going to define the groups and calculate the distance within the groups, in this case, the sibships. And then sum of all those distances and provide you with a figure that allows you to decide where you want to make the cut. So I'll explain this in a second once I get this running. It's taking a little bit of time to get the genomic matrix. Probably my computer is a bit filled up. There we are. So this is the genomic matrix, and I'm going to use the sibships. So it's going to take the GRM, perform a cluster analysis. You have options for full Sibs and half Sibs, and this is just because it's going to use a 0.5 or a 0.25 as a reference to make the groups. What it does, again, as I mentioned before, it calculates some cutoff points. Let me see, cutoff points for a cluster analysis, and says if I cut the groups up here on the cluster analysis, I'll get 3 groups. And you can see the individuals on that group have a mean relationship of 0.014 that goes from minus 0.16 to 0.7. Now what you have here is the sum of the squares of those differences, and the minimum one is the one that tends to define the best grouping. This is a bit like the nearest neighbor, but you see here a value that actually defines another cut much lower. And then let me just run that one, which actually it's in a value approximately of 1.05. And then it's going to take my cluster analysis and split it on groups, and then give me the summary statistics of these groups and we can see what happens. It found 90 sibships, which might make more sense the way we have this structure. And you can see here a few of the sibships. For example, this sibship has 276 pairs of relationships. The mean is 0.041, and there's a relationship of 0.47 to 0.61. So 0.27 is not as high as we expect for full Sibs, but it can happen for a group. And you can see there's no negative values or anything. So this is defining nice little clusters of individuals, all with a mean slightly below 0.5, which is actually a problem with the band rate and matrix. And that's it, really. So, and actually I can verify that I can get the list of these individuals. So here you have the sibship 1, and it tells you all the individuals that belong to that group. Again, most of them are going to be full Sibs. There might be a few half Sibs in there. I'm not reconstructing the pedigree because I don't have the parents. So the best I can do is say, hey, you two probably belong to a family. And it might be that even one of the fishes in here might be a male parent or female parent if you put the data from the parents in there without identify them, or it's just offsprings. So again, just giving you a little bit of flavors of things that sometimes are relevant to do here and there. And one of my last slides is where I have seen use of some sort of parental reconstruction or pedigree reconstruction where we think something like this might be relevant. The case more common for me is in aquaculture. In aquaculture, in some fish aquaculture, what you do is you perform your crosses, male, female, and you have a group of eggs that are hatching in a small little pot, but you might have hundreds or sometimes millions of little eggs hatching, and you cannot evaluate all the individuals. What you do is you take a group of those individuals, a sample of those individuals, and you put them in a common tank from every one of your families. So they are growing in a common environment, and then you avoid the tank effect. The problem is that when they grow, you don't know which ones were the parents. So what you do is you take a DNA sample, genotype the sample, and because you know which parents you use and you have your parents' genotype, you match those. Those are pretty much the example I show you with some of the salmon. You match which one are the parents, and then you have your pedigree reconstructed, and you might only use 200 SNPs, 500 SNPs, and then you have everything in there in order. So that's one of the typical cases. In forestry, we have two other cases. Sometimes we only know the female, and I mentioned this before. We collect the seeds from a seed orchard, but we are not doing controlled pollination. We know which one is the female, and we plant them into a trial. So the male is NA or 0, but some of the males are going to be there. And then what we can do is, if we have the parent's genotype, we can again reconstruct on our experiment the actual potential crosses. Okay. The same thing can be done to reconstruct some plantations where you use seeds from a greenhouse, and then you want to make sure which one they pair with what. And in ecology, this is also widely used. I've seen it with microsatellites, but if we have SNPs, it's a bit better. Sometimes you have eggs from birds, and then you have the little babies hatching, and you collect DNA from the babies, and you only know the female. You don't know which one is the male, but you might collect some males from there, and you can assess, for example, if these two kids from the female belong to the same male, or they're different males, if it has changed over time. All those kind of like sort of ecology, wildlife studies can become very useful when you do some sort of pedigree reconstruction. You don't need necessarily to go to the genomic selection, to the GWAS or base B models. Again, it's a matter of just reconstructing the pedigree and moving on with that, okay? So just one more final comments. Again, pedigree is very important. We usually have it historically, so I think it's very relevant that we, even when molecular data, we try to verify it, to check it. It's very valuable Again, I come mostly from trees. When trees, we have trials that were established. It takes a long time to get information on trials that were established historically that I'm still going to use today and probably another 20 years from now to train my models. So I need to use them, okay? Now you have strategies where you can also match the pedigree reconstruction with the genotyping. You might do the whole experiment with a low density panel for just a few dollars per sample. And once you made your selections, you move into a high density panel. And what you can do there with the low density panel, you just reconstruct the pedigree. And then with the high density individuals, you use them there into a single step or HBLAP, single step lab, GBLAP or HBLAP. So there is some options in the genotyping to do that early on or do it later. As genotyping becomes a bit cheaper, might be relevant. Now, pedigree information in some breeding programs is going to stay there, it's going to create value, and then we just need to focus on it. So reconstructing pedigree or just using QC for the pedigree might be very relevant, especially if we have molecular data. And that's it. Okay, I presented a lot of things, but I think it's been. So let me try to bring my.
Thank you, Salvador.
So we seem to have some questions. I think from Roberto. Hello, Roberto. Long time no seeing you.
Yeah, hi Salvador, good to see you. Can you hear me?
Yes, no problem.
Congratulations for your presentation. It's always good to hear something from you. So my question is related to the QC part. When you recommended like bending the matrix, the molecular matrix, using the pedigree, my question is more related like to capture the Mendelian sample. You know, when you have like a small progeny, it's a bit hard to capture the Mendelian sampling. And if you ban using the pedigree, how far you can go without like reducing even more like our ability to capture the Mendelian sampling?
So it's a good question. You're mostly talking about how much value the pedigree information has in the A matrix in relation to the G matrix. Is that right? I have done some simulations in salmon data, some not simulations with real data. And I found that when I have 700 SNPs, if I use the pedigree with the genomic matrix, I require 80% of the genomic matrix and 20% of the pedigree information. Sorry, the other way around. 80% of the information should come from the pedigree and 20% comes from the GRM, and that gives me the best likelihood value. But when I have 3,000, it was the other way around. It was 80% the genomic, but I still had 20% from the pedigree information. And I went even higher numbers because I managed to get all the way to 50,000 SNPs, I think, or 33,000 SNPs. And always the pedigree had a little bit of information. And I think it's mostly to just fix some of the errors and the instability of the sampling of the SNPs. So.
Thank you.
Welcome. Yep. Anybody else has a question?
I have a question. Is there any limitation to population size in this? For example, I have 50 parents and 350 hybrids. I'm wondering if this could still be robust for that.
If you're not using the genomic relationship matrix, there's no limitation. I mean, you can have 90 parents and 13 kids that you want to assign to potential crosses. Obviously, as you have more parents, there's more potential pairs, and then you're dealing with a very large matrix of all the possible pairs. So it's going to take longer time, but it's going to run. Now, when you're using the genomic relationship matrix for some of the checks, having a very small sample of individuals in the genomic relationship matrix usually leads to some sort of instability and bias. And that's where you have to be a little bit more careful. So the genomic relationship matrix with 200 individuals, it's okay, but it's kind of like a bit on the limit.
Thank you very much.
Problem.
Great talk.
Thank you.
Any other questions for Dr. Gizan? I have a question. So, you know, there's been in animals and somewhat in certain crop species in corn, uh, people have looked at the percent genetic variance explained, right, between markers and pedigree information, right, when you have missing levels of both, right? So there's a point at which the markers surpass the percent, essentially percent genetic variance explained beyond the completeness of the pedigree information. So I guess my question is how do you see this? I mean, is this method complementary to that, or is there a point where it makes no sense to reconstruct the pedigree when you're using it for those purposes?
So I tend to think from some of the checks that I've done that there's always a bit of information on the pedigree. At 10% or 15% always makes a correction. Now, as the breeding programs progress and we deal with genotyping of several generations, we'll get away from the pedigree and we'll have more molecular data. So in that case, I kind of envision that in 10 years from now, our last 10, 15 generations will be genotype. And in that case, we are not going to care too much about the pedigree because the GRM is telling us the relatedness, is telling us the inbreeding and so on. So in that particular case, I don't see much of a value of the pedigree. However, there is a few issues with the genomic relationship matrix that can only be corrected with some form of historical pedigree. For example, the building of inbreeding. If I'm dealing with my last generation to calculate my GRM, it's going to look at the rates of inbreeding, but that's in reality, when you think about it, is your inbreeding should be constructed from your base population, which you don't have a genotype. So a combination of the use of the A matrix that tells you this is how your inbreeding has been building, and that combined with the genomic relationship matrix to shift the genomic matrix towards the diagonal value to the average value of your pedigree based on your pedigree, it will give us a bit of more information. And again, it's going to tell me that there is a little better control on this inbreeding. Okay, so that's what I think there is still a value.
So it really depends on when you started using markers and how comprehensive that across the history of the breeding program you've utilized those, and may become a mix or just genotypic data.
Yeah, and also you also have to think that many of these sort of issues of inbreeding and relatedness are a bit less relevant when you're using something like a base B, or maybe when you use some machine learning approaches to fit a prediction model. If you're thinking from a perspective of a prediction model, it really doesn't matter. Once I'm starting to look about what is the relationship, what is the inbreeding, can I control for the relatedness and so on, then you're talking about using the GRM, which is a different story in this sense. Okay, a good question.
Wonderful. Thanks, Salvador. Well, we're at just past the hour. So if there are no other burning questions, I'd like to thank Salvador for his time and
thank you for the invitation.
Spending some time with us. Virtual applause for you.
Thank you.
And we hope to see you soon again at one of the NAPB conference meetings.
Of course. Thank you very much, and nice to see you all. Bye.
Take care. Bye, everybody.